Let's discuss question number 2, part A. Table 2.1 shows descriptions of 3 types of white blood cell. Complete table 2.1 by stating the names of these types of white blood cells. So over here we have descriptions of 3 types of white blood cells. So by reading the description, we have to name the white blood cell. So first description says that a large cell that has bean shaped or kidney shaped nucleus, it can develop into a macrophage. So the answer over here is the monocyte. We all know about monocyte. Monocyte is the white blood cell, which is a phagocyte, and it has a kidney shaped or bean shaped nucleus. And when monocyte leaves the blood and enters into the tissues, it becomes a macrophage. A cell that has a large spherical nucleus and a little cytoplasm, it responds to non-self antigens. So guys, this is lymphocytes. So you all know that lymphocytes have a very large spherical nucleus with very thin cytoplasm around the nucleus, and lymphocytes are also involved in responding to the non-self antigens. A cell that has lobed nucleus, it is phagocytic. So the cell which is the phagocyte with lobed nucleus is a neutrophil. All right, let's move on to the next part now. Part B. Dromedary camels are classified in the family Camelidae and live in desert habitats of North Africa and Asia. In these hot, dry environments, dromedary camels can lose up to 30% of their body mass from dehydration, causing their blood to become more viscous or thicker. Figure 2.1 shows a drawing of red blood cells of a dromedary camel. Figure 2.2 is a drawing of the human red blood cells. So guys, we can see the difference between the human red blood cells and the dromedary camel red blood cells. The red blood cells of the dromedary camel are, we can say, oval or elliptical, right? And they're smaller than the red blood cells of the humans. The red blood cells of the humans have a biconcave shape. They are larger than the dromedary camel red blood cells. And they're saying that the dromedary camels, they live in hot and dry environments where they can lose their 30% of the body mass from dehydration. And because of dehydration, their blood becomes viscous or thicker. Always remember this point, that whenever the blood becomes viscous or thicker, there is more resistance for the blood to flow, because viscosity is the resistance against the flow of the blood. So if the blood becomes viscous, it will flow very slowly. So for that, there's an adaptation that the dromedary camel red blood cells are smaller and they are elliptical. So the blood can flow easily without any hindrance, because these cells are very, very small. They will flow easily, even though the blood is viscous. All right. Figure 2.1 and figure 2.2 shows differences between the red blood cells of dromedary camels and the red blood cells of the humans. Suggest how these differences adapt dromedary camels for living in hot, dry environments. So as I told you, that we will say that red blood cells of dromedary camels are smaller than human red blood cells. You will also say that red blood cells of dromedary camels are red blood cells of dromedary camels are elliptical. So what will happen because of this? So this allows easier or swift flow or smooth flow of blood in dromedary camels. All right, let's move on to the next part. Part C. The llama is also classified in the family Camelidae. Llamas live in mountainous areas of South America, often at altitudes of 3500 meter or higher. As the altitude above sea level increases, the air pressure decreases. The partial pressure of oxygen in the lungs of mammals at 3500 meters is 6.4 kilopascals. So, guys, over here, basically, they're asking about the change in the partial pressure of oxygen with respect to the altitude. You all know that the altitude sickness is no more in our syllabus, but still, we have been given a question regarding that. But don't worry, the examiner has given us the information that as the altitude increases, the air pressure decreases, or we can say the partial pressure of oxygen decreases. So you should know that at high altitudes, what happens is that the partial pressure of oxygen is low. So if animals are living at high altitudes, they will have their hemoglobins will have a higher affinity for oxygen because the partial pressure of oxygen is very low at high altitudes. Are you getting my point? I'm repeating this again, that the animals which are living at high altitudes, because at high altitudes there is a lower partial pressure of oxygen, so these animals in these animals hemoglobin has a higher affinity for oxygen. So even though the partial pressure of oxygen is low, but these animals, the hemoglobin in these animals will pick up a lot of oxygen because of the higher affinity of hemoglobin for oxygen, right? So let's read it again, that the llama is also classified in the family Camelidae. Llamas live in the mountainous areas of South America, often at altitudes of 3500 meters or higher. As the altitude above sea level increases, the air pressure decreases. The partial pressure of oxygen in the lungs of the mammals at 3500 meters is 6.4 kilopascals. Figure 2.3 shows the oxygen dissociation curve of adult human hemoglobin and the adult llama hemoglobin. So we can see that the dotted line represents the oxygen dissociation curve of the adult human hemoglobin, and the firm line shows the oxygen dissociation curve of the adult llama hemoglobin. And we can see that the hemoglobin dissociation curve of the adult llama hemoglobin is towards the left. So we can say that there is higher affinity of llama hemoglobin for oxygen as compared to the human hemoglobin, right? With reference to figure 2.3, explain how the differences between the oxygen dissociation curves for the humans and the llamas show that llamas are better adapted for living at high altitudes than humans. So over here, they're asking that with reference to figure 2.3, explain how the differences between the oxygen dissociation curves for humans and llamas show that llamas are better adapted for living at high altitudes than humans. So we can over here say that the adult llama hemoglobin has a higher affinity for oxygen as compared to the adult human hemoglobin, and this can be shown by the graphs. The adult llama hemoglobin dissociation curve is towards the left. We can also quote values that, for example, if we talk about 6.4 kilopascals, so at 6.4 kilopascals, this is 6.4, at 6.4 kilopascals, the percentage saturation of adult human hemoglobin is 79%, right? Whereas the percentage saturation of adult llama hemoglobin is 86%, right? So basically adult llama hemoglobin is adapted to carry and transport oxygen at high altitudes, right? So llamas are better adapted for living at high altitudes than humans. So we can simply say that adult llama hemoglobin has a higher affinity for oxygen as compared to adult human hemoglobin. Adult llama hemoglobin has a higher affinity for oxygen as compared to adult human hemoglobin at partial pressure of oxygen of 6.4 kilopascals. The percentage saturation of hemoglobin with oxygen for llama is 86%, whereas percentage saturation of hemoglobin with oxygen for humans is 79%. So guys, there's a difference of 7%, right? There is a difference of 7% saturation. So llama hemoglobin is better adapted. So llama hemoglobin is better adapted to carry more oxygen, to carry and transport more oxygen at high altitudes or low partial pressures of oxygen. All right, let's move on to the next part. So part 2 says that sketch a curve on figure 2.3 to show the effect of an increased carbon dioxide concentration on the percentage saturation of adult hemoglobin with oxygen. So guys, we all know that as the carbon dioxide concentration increases, or we can say as the partial pressure of carbon dioxide increases, so the affinity of hemoglobin for oxygen decreases. This is Bohr's effect, right? And this will lead to Bohr's shift. So whenever partial pressure of carbon dioxide increases, what happens is that the hemoglobin dissociation curves shifts to the right, showing that there is decreased affinity. Always remember this point, that when the curve shifts to left, there is increased affinity, right? L for left, L for love. So whenever the graph shifts to the left, the love of hemoglobin or the affinity of hemoglobin for oxygen increases. And when the graph shifts to the right, that means that the affinity or love of hemoglobin for oxygen decreases. So whenever partial pressure of carbon dioxide increases, there is decrease in the affinity of hemoglobin for oxygen. This is Bohr's effect, and this leads to the Bohr shift, which is the shifting of the curve to the right side. So the adult hemoglobin or the adult human hemoglobin dissociation curve will shift to the right. Let's move on to part 3. Explain the importance of Bohr's shift in metabolically active organs such as the liver. So what is the importance of Bohr's shift in metabolically active organs such as the liver? So because liver is metabolically active, then we all know that liver is carrying out a lot of aerobic respiration, right? Liver is carrying out a lot of aerobic respiration. So liver is producing a lot of carbon dioxide. And what happens is that because of high carbon dioxide concentration, there is greater Bohr shift. The hemoglobin dissociation curve shifts to the right, decreasing the affinity of hemoglobin for oxygen. So more oxygen is released, which is used in aerobic respiration, and aerobic respiration generates ATP, and the ATP is required for other metabolic reactions, right? So what is the importance of Bohr shift in metabolically active organs? The Bohr shift allows the release of oxygen from the hemoglobin, and this oxygen can then be used in respiration to generate ATP for metabolic reactions. So over here we will write that metabolically active organs such as the liver, metabolically active, metabolically active organs such as the liver produce high concentration of carbon dioxide. Right? High concentrations of carbon dioxide causes the affinity of hemoglobin for oxygen. High concentration of carbon dioxide causes the affinity of hemoglobin for oxygen to decrease. And how? Via the formation of hemoglobinic acid. We all know that whenever carbon dioxide concentration increases, there's production of more hemoglobinic acid, and this decreases the affinity of hemoglobin for oxygen. You all know that when carbon dioxide concentration increases, what happens? More carbon dioxide combines with water, forming what? Carbonic acid. So when more carbon dioxide is there, whenever there is higher carbon dioxide concentration, more carbonic acid is produced. Then this carbonic acid dissociates into hydrogen ions and hydrogen carbonate ions. So because of higher carbon dioxide concentration, more hydrogen ions are produced, and these hydrogen ions are picked up by hemoglobin. And when hemoglobin picks up these high concentration of hydrogen ions, so hemoglobin becomes hemoglobinic acid, leaving oxygen, right? So hemoglobin normally is bound to oxygen, forming oxyhemoglobin. But whenever there are large numbers of hydrogen ions, right, hemoglobinic acid is formed and the hemoglobin releases oxygen. So this is how the affinity of hemoglobin for oxygen decreases and hemoglobin releases oxygen. So high concentration of carbon dioxide causes the affinity of hemoglobin for oxygen to decrease via the formation of hemoglobinic acid. So more oxygen is released by the hemoglobin. More oxygen is released from the hemoglobin. And the benefit or the importance of Bohr shift is that this oxygen is used in respiration to generate ATP, right? And ATP are used in other metabolic reactions. So guys, we are done with question number 2. Now we will move on to question number 3.